Brianna. Shocking, I know. And we are back on a Wednesday to talk kitchen chemistry. Woohoo. So, last week we concluded the epic saga that was sourdough bread. I think I finally vacuumed up the last of the flour. It was a time. My family ate many a carbohydrate, which is great. But after doing like a very kind of labor-intensive labor of love with Isaac Gluten and the whole sourdough bread saga, I thought it might be kind of nice to go back to the classics. We're going to talk the next couple weeks. We are going to talk some of like our classic science experiments that are really easy to do at home. It's all stuff that you have at home, I know, because I'm going to do it today with just stuff I found in my house. So that's probably what we're going to do for the next couple weeks. Today we're doing some strawberry DNA, might do some cheeky elephant's toothpaste. We'll see how it goes. So, as I just said, this week we are doing strawberry DNA extraction. So, first things first. We've talked about cells a little bit. We talked about what is a eukaryote, what is a prokaryote, that cells need, just like people, just like animals, just like plants, need nutrients so that they can make energy and survive. Yada, 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 yada. How do they do that, though? Like, how does my body know that if I get, like, a cut or something, I'm like, sorry, I'm getting a lot of double chin today, so I'll back up. Sometimes, if, like, I got a cut or something, how does my body know to fix that? Like, how does it know to make new skin, or if I like make a blood vessel or something, how does it know not to just completely fill it up with new skin? How does it know to like repair the blood vessel and fix the skin so that it like has a hair follicle again or what have you? How does it know to do that? Well, our bodies, just like every living organism on Earth, has something called DNA, which is deoxyribonucleic acid. But you don't need to know that. We call it DNA, unless you're really pretentious. But like live your best life. So DNA, there's also RNA. I was a little presumptuous. Some things use RNA, but for all intents and purposes, we're going to talk DNA today. So in me right now, there's a whole bunch of DNA. And it is the instructions exactly of how to make me. So it's what makes Brianna. They are the instructions that make my hair the way it is. It's the instructions that make my hands the way they are. It's instructions that made me the height I am. Instructions that give me freckles, make my stomach work, make me lactose intolerant, make my feet big. I don't know. They make me. And similarly, I have a plant on my table. That plant has its own DNA that tells it to be a plant. It's got DNA that tells its leaves how to work and how to feed itself and how to make roots and how to make flowers and the whole 9 yards. And the analogy I like to use for DNA is because it's a really simple molecule. It's simple and it's complex because DNA is only made up of 4 varying parts. Kind of like if we think of like computer code, how we just have like binary numbers. I say this not doing a lot about computers, but we have ones and zeros. And that's what tells those ones and zeros in different combinations tell your computer exactly how to do everything, I think. So it makes YouTube Live run for me. It tells my computer to Google how to like make rice, even though I've been making rice for a bajillion years. You gotta check. It's what helps Amazon Prime run for me so that I can order pen refills when I'm all out. It makes my computer games work, or video games, or what have you. It's the same thing with DNA. DNA has 4 base molecules, just like I said. It's got guanine, cytosine, adenine, and thymine. These 4 molecules that when put in a row in different combinations write out the very long genetic code that makes up the instructions that make me and you and your cat and your dog, your brother and your sister and your mom and your dad and your aardvark, I don't know, your houseplants, your lunch. Because everything alive has some kind of instructions inside of it that tells it how to work. So, what we're going to do today is we are going to take the DNA out some strawberries and we're going to look at it. Now, I'm in my kitchen. I don't even have a microscope here, which is one of the things that surprise people because if anyone's going to have a microscope in their kitchen, it's going to be me. And even with a microscope, we couldn't see the actual bases of the DNA. It's very, very, very small because there's like kilometers of instructions, all packed teeny tiny in your cells. Because each cell has its own copy of this genetic code. And it's not all loosey-goosey like this horrible spaghetti monster that is all tangled up. It makes no sense. It is packaged very, very neatly by your cells. They're very organized. Kind of like a library. Have all the instructions for all these things nicely organized so that when they need a specific part, they know where to go, get that little part, copy it, do what they do, and move on. So it's compacted really, really tightly, and its last bit of compacting is chromosomes. Remember how when we talked about yeast, we talked about some cells have a nucleus and some don't? Cells have a nucleus. The chromosomes and all your genetic material, that's where that lives. It's kind of what the nucleus's job is to hold all your chromosomes with all your information on them. I'm going to fire a lot of information on you, but the great thing is even though this is live, you can go back and watch it later. So when I start talking really, really fast, you can check in later. Don't worry. So, we as human beings, we are diploid creatures, which means we have 2 copies of our DNA chromosomes. Which means we have 2. It's kind of why it makes the little X shape. If you've ever seen a picture of a chromosome, this is me trying to do the interpretive dance of chromosome. We have 2 pairs. Strawberries, on the other hand, which are the topic of today's lesson, are octoploid, which means they have 8 copies. Why? Well, I did some Googling. I read some papers that were a little out of my wheelhouse. And for some reason, like at least 1 million years ago, the precursor to strawberries they had like 4 copies in each. The plants blended together, and now we have octoploid strawberries. It's kind of bizarre, and they think those are specifically the North American variety. So the kind that we grow, the kind you buy at the grocery store, are octoploid, which means that they have a lot of DNA packed inside these bad boys, which is why we use these for these kinds of experiments. Because I want you and I want me to be able to see the DNA, which is the point of this. If we couldn't see it, it would just be watch Brianna mix some things in a tube for like 20 minutes and carry on with your lives. Less entertaining. So, our strawberries are perfect for this. Other reason we use strawberries, they're squishy. I actually waited until my strawberries were a little bit off so that they'd be more mashable, because we want to break the heck out of these cells so that we can get access to their DNA. As we said, strawberries, just like the rest of plants and animals, are eukaryotes. They got that nucleus. We got to get inside that nucleus to get all the DNA out. So you can also do this. I've done this before. I think you can do it with tomatoes. Heard that through the grapevine. I haven't tried it. If you try it, let me know. You can also do this with bananas, and that is how I've done it previously. But funny story, I learned at the age of 22 that bananas aren't supposed to taste itchy. And I'm allergic to it. So it turns out I'm allergic to bananas, so I don't have any. So we're using strawberries. So what do we have on the old table today? We're going to go to table cam. I got my strawberries. I got a lot of stuff for you today. I've got a mug that's going to be a container for later. Got a mug full of water. You can use a cup if you want. I just like handles. I have a sharp knife for helping cut up our strawberries. Measuring cups. Measuring spoons. Same kind of old thing. Liquid ingredients. Water. Table salt. A little bit of dish soap. You can use whatever kind. I also have isopropyl alcohol in the freezer, but it needs to be super duper ice cold for this experiment to work. So it's gonna hang out in the freezer until just before we need it. The only piece of maybe specialty equipment you need is for measuring, because I don't have any sciencey equipment. I went, what's something you might have in your house? I have a baby syringe from from when we were kids that we used to give medication, but it's really nice because it's kind of like a little scientific equipment because it's graduated on the side, so it's got numbers, so you can suck up the exact amount you want. So, nice thing. Hold on, I have a little stash of them because they work like a charm when you're doing science at home. The other thing I'm using today that's like may or may not be something in your house, I'm using a mortar and pestle. It's basically a wooden bowl basically a metal bowl. Sorry. It's basically a marble bowl with a marble handle that just smushes stuff. But for the purposes of today, you just need some sort of smushing type object. So if I was to do this in a classroom, I was not going to give a bunch of kids stone tools to play with because I don't want things to go terrible. So if I was doing this in a classroom, really easy way to do it, I was just to take your strawberries, put them in a plastic bag, seal the plastic bag, close the ear, and have a willing child squish the living daylights out of it till you kind of have a goopy paste. And it should be kind of uniform looking. But if you're doing it at home and you don't want to use a bunch of plastic, which is fair, an eager child can be given a spoon, preferably wooden, and a bowl, and just told to mash the strawberries. Easy as pie. Nice fun activity if you've got littles at home, because they can do the squishing, whereas the older kids can do some finicky stuff. You've got to gaggle, different steps. So we are going to get these strawberries out. Now, in order to do this, you probably only need like 1 or 2 strawberries. It's normally how I give per person. But I'm going to do a few more today. Like I'm going to do more than you'd normally need because I want you to be able to see it. So I'm going to take my strawberries. I'm going to cut the tops off because I don't need them. And I'm specifically picking the gross looking ones because I cannot eat fruit in a timely manner to save my life. Pop one more of these back. Ooh, that one look. Actually, no, that one's kind of gross and squishy too. Sometimes. So I've got my strawberries in this bowl, in this mortar and pestle, and the mortar and pestle just squishes them right up. Yeah. And as we squish them, we're going to eventually transfer everything to this container, which is going to have a sieve over top, which I realize upon reflection isn't the cleanest, and also isn't very balanceable. So as we do this, we're going to pour some of the juice through. Yeah, there it goes, because I probably added too much to the mortar and pestle. So, sorry, forgive my younger brother who is giggling in the background watching something on YouTube. Asked him to go to his room, but here we are. Pour, smoosh, and pour, and smoosh, and pour, smoosh and pour, smoosh and pour. And we want to keep doing this until this is as much of a uniform paste as we can get. Whenever it looks like it's going to overflow, we pour. So there are some things that aren't going to pulverize super well, like seeds, for example. That's okay. That's why we have the strainer, because we don't really want chunks, because that's going to kind of interfere with our experiment. Keep smushing. Sometimes if you're using a mortar and pestle, especially one like this, where like the thingy's big, you might need to do a little bit of scraping, which is why I have a knife. This is my obligatory safety announcement. Don't use, if you're using a knife or pointy implements, please be safe and use them with adult supervision if you are on the younger side, because even though I'm an adult, I have sliced myself a few times. My brother is continuing to laugh. I'm going to pretend he's laughing with me and not at me. So I'm going to keep smushing. Quality television, right? Smushing strawberries. Just keep smushing them. Smushing them nice and good. Alternatively, you can also, once you get it like pretty squished, your mortar and pestle. Ugh. That was one of those ones, folks, where you're going to get steaky. Now scoop that out. And then I'm just going to grab a spoon because I think that's going to make this a little bit easier. Just a little bit. So I'm going to give myself, that's just dripping. Rinse my hands because they're sticky. Ultimate piece of scientific equipment, a spoon. So you can take your strawberry goobs and you can squish them on the side of your sieve through your spoon. See, this is one of those things where, this is why I write my instructions afterwards, because often I'm like, this is how this activity is going to work. This is how I do it in the classroom. And then suddenly I have to transfer it to home. And maybe if I do it in a classroom, it can be done more easily at home or a different way. So that is why sometimes the instructions are a little bit later, because I like No matter how many times I test run it, I always sometimes find a better, different way to do it. And if you ever find a better way to do it, and you're like, hey, Brianna, what are you talking about? Try it this way. Let me know, post in the comments, and I'll be like, heck yeah, better way to do things. I love finding better, easier ways to do things. It's kind of my goal in life. Okay, I know, riveting YouTube. I think that's as sploogey as we're gonna get. You know, technical term, sploogey. Can't take credit for that word. I used to work in a kitchen, and anything that was kind of gross, the chef called sploogey. So you see we have this nice kind of juice along the bottom. I'm gonna scrape that into our container. Normally we use a test tube for this, but I did not have a test tube because, well, it's a kitchen. Oops, I lost some. You know, sacrifices sometimes have to be made in the sake of science. Okay, I got a nice amount of strawberry goob. So I'm gonna move, I'm gonna rinse, I'm gonna dry with the tea towel, and we're back. So we gave that a good rinse. I have some washing up to do later. Ooh, said I was gonna do more, didn't I? Okay, I'll do 2 more and I will explain the next steps. Back comes the sieve, and we shall carry on with our merry adventure. So, there are 3 things that are going to help us get our strawberry DNA extracted today. And they are isopropyl alcohol, salt, and soap. And this is actually pretty close to what we use in the lab. We just call it technical words like, well, we use different salts sometimes. We call it a detergent or some other fancy alcohol, and we freeze it at fancy temperatures and blah, 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 blah. It's the same idea. This is what I learned how to do in university, but just more simple, using household things. So, we already talked about that the DNA is in our cells, but it is kind of tucked away in those, it's tied up in those chromosomes and it's tucked away in the nucleus. So how do we get it out of the cell? We can bash the living daylights out of it, but cells are pretty small. So just by doing this isn't going to have me break up all of the cells. So how are we going to do that? Well, this is where the soap comes in. So soap being soap, what it does is it helps break down fats. That's why if you've got like greasy hands, like let's say you have a real good burger and you're washing your hands afterwards and you give it a little soapy rinse and you still can't get it off. This is why we use things like soap to get grease off, because it is really good at breaking those bonds between fats, which allows the fats to be rinsed away. Kind of works, there's similar kind of things in your digestive system, because otherwise, every time you eat a fat, just glob together in your digestive system, it wouldn't, I'm doing it onto the counter. It would just glob up in your digestive system and you would just excrete, let us say, fat. Isn't great. That's what happens to your gallbladder, doesn't work. So what we use is we have chemicals in our body that work a lot like soap, again, to break down those fats. So that's why we use the soap, because our cell, like layers of cells, often have lipids in them, which is a fancy science word for fats, to help keep water out because they don't like mixing together. So by adding the soap, that helps break down the outsides of the cells, cell wall and the like, so that we can get at that good DNA inside. So, second thing, salt. What's the deal with salt? So the salt helps with interacting with the proteins that kind of hold the DNA together. Like how I said, the DNA is like nicely organized. It's organized in nice little patterns. Ooh, we're getting more. That is, it's helped together with proteins. It's kind of like beads on a string. Anything that keeps the beads on the string, kind of like the proteins. So salt added helps disrupt those proteins to make the DNA like loose in your solution. And I also think it also helps make the cells more permeable to stuff like alcohol. So the alcohol is another part. So normally DNA, like strawberries, they're wet. They're wet because they're filled with water, just like people. A lot of organic life forms made of water. Most organic life forms largely made of water. A lot of water. So lots of things, like biological things, are able to dissolve in water. Fats aren't. Just like how we kind of said with the digestive system, and again, that's why we need the soap to rinse it off. Alcohol, on the other hand, the DNA can't dissolve in the alcohol. It kind of bundles up into chunks, and it'll float on the top. So where everything else will sink to the bottom. I just got strawberry in my hair. Isn't that nice? So, to recap, we got some good juice. Good juice. I'll show you in a second so I don't spill it everywhere. Da, 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 da. Singing with Rihanna. There we go. So, let me rinse so I don't put more strawberries in my hair. So, when we add the soap, helps breaks up the cells, releases that DNA. Salt helps break up the proteins that are holding the DNA together, so it's sort of loose in our solution. And then the ice cold alcohol helps the DNA to separate from the solution. And this is called precipitating. Precipitating just means when we add something to a solution and it causes a part of that solution to separate out. So sometimes if we want to isolate something from a mixture, like let's say we want to get caffeine from coffee or we want to get caffeine from tea. We'd want to precipitate out that caffeine. We'd want to separate it from the solution that we put it in, just like how we're doing today with our strawberries. So, first thing I'm going to do, it's nice and dark. Like we had a couple of seeds get through, but we try our best. Again, we're doing this in the kitchen, not a lab. I like food science because I can eat it. So I am going to add some water just to dilute our solution a little bit. I'm going to give this a good stir with a knife because that is what I have on hand. And add a little more water. So we have this nice, also kind of get some of the strawberry goop off the sides. Strawberry goop. No strawberry goop. There we go. So this nice solution of strawberries and water. It's delicious. Don't eat it after we add the soap. So now we are going to add a healthy amount of soap. And the good thing about this is I will give you written instructions it'll be a link to the video, probably sometime by the end of the week. We link to the video, written instructions for how much to add if you're using 2 strawberries. But the good thing about this is kind of the more the merrier. The more soap you add, the more you're going to break down the cell wall. The more salt you add, the more it's going to disrupt the protein interactions. The more alcohol you add, the more it's going to precipitate out. So for all intents and purposes, because I've done this before, I'm going to eyeball it, but there will be written instructions for you folks who don't do this as a job. So I'm going to add a tablespoon of soap to start, because that seems like a healthy amount. And I'm going to give that a good stir with my measuring spoon. You want this nice and incorporated. The stirring is important because you need those cell walls to be nice and broken up. So we can see it's a little frothy. It's a good sign. Okay, awesome. Now we're going to add our salt. Can you tell I eat a lot of the same jam? And I'm going to add, I think I'm going to add 2 teaspoons actually. Help break up those protein interactions. Protein interactions. I'm going to give it a good stir because again, we want that salt to interact with our proteins along with the soap to break down those cells. So, you can see now. Now, a lot of this is bubbles, but it has gone cloudy, and that's partially, besides bubbles from the soap, partially because our cells are broken up. So instead of it being clear, instead of it being like water, and then the water from the strawberries and cell solutions, it's not just floaties, we've broken them up. So there's a lot of little pieces drifting around now, which is exactly what we wanted. So kudos, we're doing great. This is the part. If we're going to have any tricky parts in this is the part. And even then, it's not that tricky. So, isopropyl alcohol works best if it's like stone cold. You want it as cold as possible. I put it in my freezer like 3 hours ago. It doesn't freeze because it's alcohol. And it's just rubbing alcohol. Like it's what you have it cut, what you use to disinfect stuff. That's all it is. And this is why we also have the baby syringe. Because, I forgot to take the top off. Because we want to dribble it gently down the side. If we just shoot it in, it's going to mix in with our solution. Whereas if we gently dribble it down the side of the cup, it's going to float in a layer on top. And that's what we want, because if it's all mixed into our solution, we're not going to see that precipitate form. But if it's floating on top, because alcohol is less dense than water, the water kind of sinks underneath it, and floating on top, it'll let the DNA precipitate out. So what I'm going to do, and I'm going to do this speedy, to get the alcohol out, I'm going to pour it in this mug so that it's easy to suck up. For me, and I'm going to gently dribble it down the side. I may have my concentration face on, so if I'm not talking, that's why I'm talking beforehand, because I gotta be efficient. And shockingly, I'm not the most efficient human being on the face of the earth. So, we got the mug cam. There we go. Isopropyl alcohol, frozen, shaken, not stirred. Adding it to the cup. Put it back in the freezer and do something more. We get the baby syringe. We suck up some alcohol. And we gently, so put it up to the side. Put it up to the side this way, and we gently dribble it down the side. It's going to be tricky for you folks to see. On your side, you can see it spreading because it's popping the bubbles. It is gently forming a layer on top. And again, I will give you exact amounts, but I am just, because I'm using a really wide cup, normally I'd use something like a test tube, but there aren't normally test tubes in kitchens. I'm just going to keep adding it until you can see it. So again, holding the side, gentle, gentle, gentle. Depressing the plunger. And getting that nice layer of isopropyl alcohol on the top. I'm going to probably do one more. And each of these baby syringes is about 2 tisps. So for my cup, I'm probably going to do 6 tisps or teaspoons. This is probably an adult step you want to do because isopropyl alcohol is not safe to drink and it is flammable. So we're not using fire today. But this is a good step to maybe ask an adult for help or provide your youth in question with some supervision. Okay, sweet. Got strawberry juice everywhere. But that's fine. So I'm going to very carefully hold this up so you can hopefully see the layers. So we see, oh, actually it's already doing it. Woohoo! So you can see we have our strawberry goop. It is kind of separating out. And that's our layer of isopropyl alcohol on the top. And do you see that kind of snotty stuff floating in there? That snot is our DNA. It's globbing together, kind of like how I talked about fats glob together, because again, they don't dissolve in the alcohol. So they have kind of globbed together in this snotty mass in our alcohol. And there's like some good globs I can see on the side. The longer you let this sit, the more it's going to precipitate out. I don't really think that you want to watch me wait for this for 10 minutes. So maybe I'll post like an updated pic or something like that on our Instagram. I'll show you when it's all done. But that's all you need to do. It's pretty straightforward, pretty easy. It's all using household stuff. You don't have to buy any of this from the store. Again, you can use other fruits and feel free. Try other fruits. Again, I chose strawberries because they were in my fridge. Try tomatoes, try bananas, give it a try. The key point is just try to get as much juice out of it as possible. Use a sieve, separate it out, and then follow the regular steps just like I showed you. Again, written instructions should be up by the end of the week. Again, I like tweaking it as we go, as I try it. So, thank you so much for joining me on this week's Kitchen Chemistry. we are going to continue our series on our classic kitchen experiments for the next couple weeks. I hope you enjoyed it. If you have any questions, comments, concerns, things you want to share, things you want to chat about, hit me in the comments, talk to us on Instagram, contact us on our website. I want to hear from you. So, I'll show you one more because it's kind of glued down some more. Whoop. Yeah, so a lot more snot that time, hey? Look at that snot. Good DNA snot. Can you tell I like science? Thanks for joining me so much this week, folks. I will see you next Wednesday for our next episode of Kitchen Chemistry. Bye!